Okay, so thanks organizers for inviting me for this symposium in the honor of Professor Madhavan. So this picture is from Gordon Conference in year 2002, and you can see this is 24 years ago. So yeah, so Professor Madhavan is here, and myself is here. So about 6 to 7 months after this conference, I joined Professor Madhavan's group in Cambridge. And then, of course, he moved to Harvard. So when he was in Cambridge at that time, he brought his lab's floor plan and asked me to ask that, tell me where you can put which equipment and where the bench, working bench. So I designed his, So anyway, now he has a different lab, I know. So after coming back to India, I set up this soft matter laboratory. His laboratory, of course, soft mathematics laboratory. So these are some pictures of my students over the years. So here I have worked quite extensively on soft materials, gels, elastomers, interfaces, weighting, instabilities. This is elastocapillary effect, interaction of soft, solid and liquid, and many other material, many other problems. I will say that many of these problems are actually, the idea, of course, is there, but the way that we thought process, I think, has been affected by, influenced by Professor Madhavan. So thank you for always being there. So today I'm going to talk about this particular problem. This is confinement-induced alteration in surface energy and consequent changes of morphologies of complex emulsion. So these are done by my students, these 3 students. So it's a MTech student who just graduated from Cornell, PhD, as a PhD student, and Orko is currently in Texas. So emulsions, we all know, we see them in pharmaceuticals, foods, cosmetics, and paints, and like that. So in a sense, like you have got, let's say, water and toluene in the a polar and non-polar liquid. And if you do not use any stabilizer, you try to disperse them, they will actually phase separate. But if you use a surfactant and you stabilize it, and if you look at it under microscope, it will look something like that. So here, basically, there are 2 different liquids, one water, another let's say the oil. But we have these complex emulsions, in which case you have got basically 3 liquids. So one is this blue, one is this outer liquid, and then there is this coarse shell, and basically this particular, in this case, there is a liquid that remains engulfed by another liquid, let's say. And then there is another particular form, this is called genus. So basically 2 different liquids, they remain joined. Both the liquids face this outer liquid, okay? And then there are many other complex morphologies having like maybe more than 3 liquids and like that. And they have their, applications. There can be many applications. So some of them are here published by, published papers from different groups and like that. And so what we looked at is something like this, that how this morphologies that change. So one is to apply some stimulus. So let's say that this is the morphology and coarse shell morphology to genus. And normally these are the different types of stimulus that people have used: photoresponse, temperature, binding complexation, in situ surfactant formations, or cleavable surfactant. So surfactant is there, so it exposes light, so it cleaves, and then that way the surface energy changes and the morphology changes. And then there is pH response and like that. So basically one can think of something like this, that this is A is the inner liquid, B is the outer liquid, and I is, of course, the environment, let's say the water. And so when will you see this coarse shell or genus, and let's say the reverse coarse shell? So the conditions can be find out by considering this, that suppose a liquid drop on a surface, on a substrate. So this is, let's say, A and this is B. And so the surface energy here is gamma AI. So gamma AI is strong enough that it pulls this three-phase contact line. And so one can define what is known as the spreading coefficient, defined as the surface tension of A, interfacial tension between A and I. This is I is the outer liquid or outer phase. And then there is gamma BI and gamma AB. So these are different interfacial tensions. So one can find out the spreading coefficient. So if the spreading coefficient is positive, so which means is that gamma I is quite strong, so it will actually pull this liquid B over it, and it will completely spread. So now if this surface A, instead of becoming a flat surface, if it is a curved surface, okay, then this B is going to completely engulf A. So that way one gets this particular kind of equation, that where delta AB, which is defined as gamma AI minus gamma BI, is greater than gamma AB, then we get this core shell type of morphology, where A is inside and B is outside. On the other hand, if gamma B is greater than gamma A, then of course one gets this kind of morphology, which is also a core shell, but here B is inside. And when this gamma AB is larger than this delta AB, then one gets this genus morphology. So it's like that. So what it means is that by altering these interfacial tensions, okay, gamma AI, gamma BI, okay, it is possible to change from one morphology to another. So for example, so here we have got like A is paraffin oil and B is silicon oil, and outside this is basically very dilute solution of agarose. So 0.1 millimolar or something like that. And at that situation, the gamma AI is this value, 32.74 or like that. Gamma BI is this. So gamma AB is something like. So these interfacial tensions can be calculated or estimated or can be experimentally measured. So that can be done. And so you can see here that this gamma delta AB, that is comes out to be 1.49. So which is actually larger than 1 here. So therefore, one will get this core shell morphology. On the other hand, in this paraffin oil, you mix a little bit of hexadecan, whereas such that this gamma AI now increases to 36. So I is basically water, or this aqua solution of agarose. So here, this gamma AI is 35.97, and however, gamma BI remains same, because gamma BI is, we do not change anything with the B, this is silicon oil. So the gamma AB is now this quantity. And so now you see that this gamma AB, which is 9.66, is larger than this delta AB. So one gets this genus droplet. So it works quite nicely. So this is also volume independent. So I want to emphasize that this is a purely surface tension driven phenomena, that when you increase this, let's say the ratio of this paraffin oil and silicon oil from 2:1 to 1.1:5. So basically you are increasing the paraffin oil quantity, and so the inner liquid drop, the volume that increases, and we get a the outer shell that appear more thinner and thinner basically. So so basically the change in volume does not affect this morphologies, different complex emulsion morphology, and also these are long-term stable. So here this is this the core shell morphology. So we kept it more than let's say 1.5 months. So the morphologies remain doesn't change, so remains same, remains stable like that. So this morphologies do not depend upon, there is no aging effect or anything, so doesn't depend upon time. So what we observed is the confinement. So when we confine this emulsion between two parallel plates, then something interesting happens. So here we have these two glass plates, and these are like wetting surface. So this outer this water or this aqua solution of this agarose, okay, is placed a liquid drop between these two parallel plates, and then you observe from the top that how does this morphologies look like, and we can actually vary the gap between these two plates, that is H. And so when the gap is about 138 microns, then as usual, this 138 is a core shell morphology. That is what is expected, and we see that. And now let's say we decrease the gap from 138 to let's say 92 microns. So still these droplets are all core shell. And by the way, this so we put in water this aqueous solution of this agarose, both this silicon oil and paraffin oil, and agitate it. So that is the way we make this emulsion. And since there is no other control, so the droplets are all of various size ranges. I mean, like very small drops to very large are there. And if we see it microscope very even with large magnification, we will see that these are all core shell. I mean, like there are no genus there, so all core shell actually. So now we decrease this gap from 92 to let's say 46 micron, and we see that this genus droplet, the core shell now transforms to this genus morphology. Okay, so as you can see that this happens without any alteration of, without any addition of any surfactant or any other stimulus. So this here, this gap, this decreased to 46 micron. So the question is that, is it because that we have sandwiched these droplets? But actually, like if you see this particular droplet, which has size of maybe a 10 to 20 micron, so that does not get, it's not going to get affected by this, by the two plates being very close. So because if the size is too small, so that also alters to genus. And the droplets doesn't this transition from this core shell to genus doesn't have any particular direction. I mean, like it happens in all different ways, randomly. So no issues there. And we can also find out that which one is silicon and which is paraffin oil, because what we can do is that silicon oil, we can, instead of using just, there are cross-linkable silicon oil that can be used. So one can actually cross-link this at this particular state, and one can get these different particles. One can actually get particles, okay? One can make this core shell or this kind of genus particles by cross-linking it, and we can find out that we know which one is which liquid. That can be done actually. And here, so so when this phenomena this happens, so we try to capture it, that how exactly this goes on. So here, this inner droplet, initially it was at the center. Now it comes to the edge, and it forms basically a thin shell, and then it tries to initially spread, and then there is a rupture that happens. So it's a kind of thin film rupture. Okay, that way this happens, and this core shell gets transformed to genus. I will come to that. So these are some of those particles that form. So in the core shell case, okay, one can get this nice particle, yeah. And here it is like genus. So it has got this curvature because this part was the silicon and the outer part was the paraffin. So paraffin oil doesn't cross-link, so it goes out. So generality of the phenomena, so is it only because of the agarose that happens? So which altered the surfactants, okay, of different surfactants, and is not the very high concentration, no, low concentration like that. 1 millimolar, 0.01 millimolar, 0.001 AOT, blah blah. And each case, so there is initially, okay, the surface energies are such that it should form core shell, but as we confine it, it converts to genus beyond some critical gap and like that. So we try to understand that what may be going on here. So initially, of course, this is at the center, and this. So when you confine it, before you confine it, the outer liquid, okay, there is no meniscus. It is almost like flat. So as a result, the pressure, Laplace pressure, is basically zero across this interface. So the pressure in the liquid phase over here is very similar to this atmospheric pressure. So this Pi, okay, is basically P atmosphere. However, there is this B liquid is there, so there is a Laplace pressure across this interface. So that can be obtained. So this is PB2 minus Pi So that is equal to 2 gamma bi by Rb. So Rb is the radius of curvature of this outer shell. And then this is Pa and minus Pb1 that can be obtained right then. This is the Laplace pressure across this interface between A and B. So like that. So now Pa1 and Pb1 and Pb2, they are same. So it remains concentric and symmetric. But now let's say that we confine it. So when you confine, there is this curvature that appears. So because of the curvature, the pressure inside here decreases from atmosphere. So the Pi is no longer P atmosphere, but it is P atmosphere minus 2 gamma by H. So the pressure decreases. So as a result, the Pb1 continuous phase, so this Pi becomes now this, so Pb2, okay, so Pb2 is over here, Pb2 is over here. So here the pressure now is decreases to some extent because the Pb2 now is actually this this P atmosphere plus this quantity minus this quantity. So pressure Pb2 actually has decreased. So initially it was Pb1 is equal to Pb2, but now it has decreased from Pb2 has actually decreased. So as a result, there is a pressure gradient that develops. So this pressure gradient is something like this. So yeah, 0, yeah, so it can be found. So it comes out to be negative over here because it as we go from inner to the outer, okay, the pressure decreases. And one can also write down the del Pb by del theta, so along the theta direction by this way. And one can actually see here that this Pb theta by del Pb theta by del theta, that is expected to be negative. Okay, so what it means is that as theta increases, okay, the pressure actually decreases. So as a result, see, if there is some perturbation, this particular liquid should go flow out from this minimum thickness region, and indeed this should become unstable, and this particular drop, inner drop, should come to the edge. So yeah, now so the growth of perturbation from concentric morphology, okay, it's a decrease in minimum shell thickness that should happen actually. So now reversibility of this particular phenomena. So before we rupture it, it is actually completely reversible. So that we checked here. So it is 93 micron. This is 65 micron. 65 micron. There is the rupture of this thin film that has not yet happened. Okay. So it has only this particular inner drop that has only gone to the edges. And now if we increase the gap once again, so it completely reverses back. However, if we rupture it completely, so the way that it happens, this becomes irreversible, apparently irreversible. So here, for example, the experiment is done not between two parallel plates, but at an angle, inclined plates. And so as we move this spacer up and down, we can actually increase or decrease the gap. So as you can see at this particular location, the thickness varies from 93 to 55 micron. So here it becomes a coarse shell to genus, and after that it remains. After that, as we reverse the cycle, okay, from genus to coarse shell, no, it doesn't go back. So apparently it appears that it is irreversible. But as you might imagine, that in order that from genus to once again go back to coarse shell, okay, it will one of the liquid has to engulf the completely weight. So that's a kind of nucleation phenomenon. So it requires an energy barrier to happen. So what my student did is, so it is a very small amount of liquid though, but my student did this experiment many times and then scrapped the liquid, so which had now all the genus droplets, and then agitated it, okay, and indeed that the genus drop once again going back to the portion. So there is this engulfing thing requires some amount of energy barrier, and once that energy barrier is crossed, this morphology goes back to coarse shell. So now the question is that what happens? Maybe what was there? So what is maybe? So now till the slides are actually my slides, and now the slides are prepared by my students. So all kinds of animations will come here. So so the question is that what is going on at this interfaces. So we are interested to know about this silicon oil outer liquid and this aqueous surfactant solution. So we devised another experiment. So in this case, we have this glass plate with a cavity, and we put a small drop of this silicon drop, and then we cross-link it slightly. Okay, slightly cross-linked it because we wanted to, we cross-linkable silicon. Okay, and then we put this inside, submerged it in a colloidal suspension of particles of 5 to 15 micron, very small particles, and then we basically confine this. So what happens is that once we do not confine the unconfined state, and this is basically the confined state. So what happens is that these colloidal particles, okay, they have this energetically, they have the possibility of coming and colonizing on this particular surface of this silicon oil, and then we cross-link it completely. And when you cross-link it and then look at it in ACM, let's say, we'll see these particles embedded to it. So the idea is that how are these particles embedded, okay, inside this cross-linked silicon. That was basically the idea, and unconfined state and the confined state. So, so what happens is interesting. So when we have this particle, so only one unconfined state, so basically there is some theory to show also that it is energetically favorable for the particles to go and colonize. So so what happens is when you have this particles, the particles actually get embedded. One can see here. Now, unconfined state, the particles are over here, something like this, and this column is for confined state. So as you can see that if it is only water, no surfactant, okay, then the unconfined state and the confined state, there is no difference between these particles, the way to the extent that particles get embedded. But when you have a surfactant, okay, and then the unconfined state, the particles are embedded only up to this extent, but the extent to which the particles get embedded, that actually increases quite significantly, right? So it is 1 millimolar SDS. This is an anionic surfactant, and so that doesn't depend upon whether the surfactant is anionic or cationic. So something happens on the surface of this silicon oil, okay, when you confine it or unconfine it. So impedance of surfactant, the particles get entrapped to a greater extent of silicon surface. So so now we are interested to know that what are the interfacial energies, okay? That if so, is it that the silicon oil water interfacial energy is increased? So in order to do that, we actually analyze these pictures. Now, if we consider that this particle is embedded, and we just draw a line and then they use this Young's equation, that will be a wrong idea. That will actually give a wrong values, because this particle, when it is embedded, the silicon after cross-linking, this does not form a smooth surface. So there is a curvature there. So this curvature and the corresponding, like the tangent, has to be drawn properly, right? So we try to do that, something like this. So basically one has to consider the Neumann's triangle or things like that, and one can find out what will be this interfacial energies. So actually we do not have the actual picture that how this from the side view, okay, of this like this. So we have only picture like this. So that whatever I am presenting here is an approximate analysis we could do, and we try to find out from this looking at someone wrote approximate that what happens to these interfacial energies between this silicon and the water over here from these contact angle measurements. So what it gives rise that when you have this 1 millimolar, let's say, surfactant solution, the interfacial energy between silicon oil and aqueous, that indeed increases from 27.8 to 48. here it increased from 33 to 46. So what it means is that the interfacial energies between silicon oil and water, that actually is getting increased because of this confinement. So, if the surface of So now above. Now what happens if we have from this consideration, that what would happen if the surfactant is the concentration increased beyond CMC? So here, if it is below CMC, when we have the pressure decreases, then there is a desorption of this adsorbed surfactants molecules from the silicon surface to the water. So that would happen. But if it is above CMC, then because of these micelles, okay, act as a reservoir for the surfactant molecules. So this molecules, the desorption is not that much expected. So that means that whatever is the interfacial energy, that is expected to remain same above CMC. So above CMC, this kind of transitions are not expected. So I will go to this experiment. So here, therefore, of course, this is core shell, and then we actually increase this, use the surfactants, which was about like 10 millimolar, so which is actually larger than the CMC value. So in this case, between the unconfined and confined, there is no change in the morphology. So what it means is that it is a surfactant molecules which gets dissolved from the silicon oil, silicon oil surface, and that leads to this increase in the interfacial energy. So desorption of the surfactant molecules from the silicon oil, that leads to this desorption of this, the increase in the interfacial energy, and when the interfacial energy increases, okay, then these particles get adsorbed more. So basically that is what we understood, and so we could also verify it by this measurement or estimation of these interfacial energies for the liquid drop case as well. So basically this is the summary, so thank you.
Thank you, Animangsu. Questions? Can I ask a dynamics question? You focus mostly on understanding it energetically, but when you switch from the Janus to the core shell, how quickly or how slowly does that happen? And you mentioned the presence of a barrier. Is there a way in which you can characterize that transition experimentally or otherwise?
So actually this, there are like two time scales. For the one is that the inner drop coming to the edge of this particular outer shell, and then the rupture of this. So the rupture follows that thin film instability like the time scale. So that we check. And the inner one, of course, also it's not instantaneous. It has its own time. It takes some time to come. That is there.
Can I ask a related question? In many biological systems, you have phagocytosis, which does not look so different from what you're showing.
Right.
Is there a way in which what you study can be used to understand active versions of what you're saying, which might respond or correspond to phagocytosis?
So it may be possible because see here we did not use surfactants. Initial thing we actually observed with agarose. So agarose is a biomolecule. So in the case of this biology, we are not going to use a surfactant, but you have this kind of long chain molecules with having this of course polar and non-polar groups and like that. So what is interesting here, it is not of course this phenomena, but over and above that even in presence of those biomolecules, okay, you can have this kind of effect, and this agarose-like biomolecule can give stability to this, this oil-water systems and like that. So I think that, and of course in the morning also there were some talks on this compartmentalization. So in some of the things, yeah, there it happens. Yes, but as far as this. How to characterize or estimate the energy barrier. So so far we have not been able to do so. We have to think about it actually.
More questions? Okay, so in that case, I just thank Professor Animangsu.